Hello everyone, it's a great honor to share our team's latest research published in iMeta. SOSP uncovers pervasive SOS-independent prophages with distinct genomic and host traits in the bacterial genomes. Prophages refer to the dormant form of temperate phages during the lysogenic cycle. They can be induced into an active state in response to environmental changes, leading temperate phages to enter the lytic cycle. This process is referred to the lysogenic lytic switch. The classical lysogenic lytic switch relies on the bacterial SOS pathway, which is activated in response to DNA-damaging agents. However, recently SOS pathway-independent induction of prophages has been reported. Furthermore, induction experiments using MMC on environmental microorganisms have shown highly variable induction rates, with widespread insensitivity. Nevertheless, due to the lack of specialized tools for determining prophage induction modes, the distribution proportions and genomic characteristics of SdPs remain largely unexplored. Consequently, developing universal tools to characterize phage induction modes is essential for systematically elucidating the biological traits of temperate phages. In this study, we develop a novel bioinformatics tool SOSP that predicts prophages induction modes, identify 11,806 SdPs by applying SOSP to 49,333 complete bacterial genomes, uncover that SdPs and SdPs exhibit distinct genomic and host traits, refine the conventional understanding of temperate phage induction mechanisms, and provide novel tools and insights for exploring the lysogenic lytic switch of phages. Previous studies have utilized the Heterology Index to assess the binding affinity between the LexA protein and the SOS box. Inspired by this, we propose the development of a tool named SOSP based on HI to determine whether prophage induction is dependent on the SOS pathway. To validate the reliability of HI in predicting binding, we first calculated the HI values for all potential SOS boxes in E. coli K12 and determined an HI threshold through clustering analysis using the mean shift statistical method. The comparison with the experimentally determined LexA DNA binding data reported by Lewis et al. Revealed a prediction accuracy of 100%, demonstrating that HI can reliably predict the potential of LexA binding. To evaluate the accuracy of SOSP in classifying prophages, we collected prophages with existing experimental data, including 10 prophages. SOSP achieved 100% sensitivity and 100% specificity in the test set. In addition, the prediction results in the deep-sea-derived bacteria Xiaonella WP3 and WP2 were also supported by experimental evidence, indicating that SOSP can accurately identify whether prophage induction is dependent on the SOS response. To understand the distribution of SdPs and SdPs in bacterial genomes, we applied SOSP to analyze 49,333 bacteria with complete genome sequences from the NCBI RefSeq database. We found 16,365 bacteria harbored CSBs. Among the prophages identified by SOSP, 11,806 were classified as SdPs. Notably, within Gammaproteobacteria, SdPs were found widely distributed across multiple genera. In certain genera, their proportion exceeded 60%. Additionally, bacterial genomes without CSB and LexA can't be predicted by SOSP. Subsequently, we analyzed the taxonomic and genomic characteristics of SdPs and SdPs to delineate the features of these two prophage groups. Overall, SdPs and SdPs exhibit both distinct clustering and overlapping patterns. At different taxonomic levels, the degree of shared features between SdPs and SdPs increased with higher taxonomic ranks. Besides, differences were also observed between the hosts of SdPs and SdPs. In summary, these findings highlight the unique genomic features of SdPs and SdPs, underscoring their distinct evolutionary trajectories and ecological preferences. Finally, this is the summary of our work. We welcome everyone to use our tool, SOSP, and kindly provide feedback and suggestions. Thank you all.